Okay, now I'd like to start Rosa Beddington Lecture. So let me introduce myself first. So my name is Yumiko Saga, and I'm one of the organizers of mouse molecular genetics. I will chair this session. So I think that many of you know what the Rosa Beddington Lecture is, but since this year has a special organization, so I like to introduce and remind about Rosa. So Rosa is a person who demonstrated that node is the organizer in mouse embryo, and the anterior visceral endoderm is important for AP patterning before the formation of the primitive streak. And she published many important papers, and those appear in textbook. And in addition, she had been known as a talented artist, and she designed the Waddington Medal, a famous award for developmental biologists. And Rosa herself received this medal in 1999. But unfortunately, she passed away from cancer at 2001. But because of enormous contribution to Mouse Genetic Society, the MMG meeting decided to have a lecture in her name since 2002. So now the time to introduce Kat. So this is my great pleasure to introduce Kat from the Memorial Sloan Kettering Cancer Center. I briefly introduce her career. She received her PhD from Imperial College London in 1995, and I heard that she was influenced a lot by Rosa when she was in London. But she decided to move to Toronto to work with Janet Rosen, and she moved to New York, Columbia. And at 2004, she was appointed as a young PI in Memorial Sloan Kettering. Since then, she has been a leading researcher in early embryo genesis. So, okay. The seeing is believing. I just put the Japanese that not so many people realize. So, but I guess this is the right word to describe her science. As you know, imaging technology quickly advanced, but it is still very difficult to observe developing mouse embryo. So she has developed several tools to address the question how initial cellular specification occurs in situ. I can recall Rosa's first trial to live imaging of AVE cell. So I think that she is the most fitting speaker for the Rosa Beddington Lecture today. So I like to, call Kat.
So thank you, Yumiko, for the very nice introduction, and I'd like to start by thanking the organizers for this really great honor. And I wanted to say, actually, my history with Rosa was that I'd arranged a postdoc with Rosa while still a graduate student in London. And that meant that I was actually going to relocate to Edinburgh. Rosa at the time was at the Centre for Genome Research. And then, unfortunately for me, she decided to relocate her lab to the National Institute for Medical Research. And really feeling that I wanted to have the opportunity to leave London, I decided to forsake that postdoc opportunity and go elsewhere. So I ended up in North America, first with Janet Rossent and Andros Nagy, and thereafter with Ginny Papayannou, all, of course, individuals that had been intimately involved in the training of Rosa and sort of collaborations with Rosa. So I think that's, really for me as a graduate student and also as a postdoc, coming to meetings such as the British Society for Developmental Biology meeting and the Mouse Molecular Genetics meeting, I felt that Rosa was a very prominent and often very outspoken feature of those meetings. And I think many of us remember her sitting in the back or at the front and asking very provocative questions, or chairing sessions and whisking speakers that have gone over their time off the stage. But I feel ultimately that her most enduring sort of a contribution to the field of mouse and stem cell biology is in her trainees, and I feel really honored to be talking in the same session as, for example, Josh Brinkman, a former postdoc of hers. And there are other former trainees of hers. For example, Tristan Rodriguez was talking at a workshop this morning, and Ruth Arkell has traveled all the way from Canberra in Australia to attend this meeting. So I feel very humbled. So what I thought I'd do in my talk is really start with the object of sort of my affections, I think Rosa's affections, and the affections of many of us here, namely the mouse embryo. And this is effectively a ventral view of an 8.5-day or an 8-day mouse embryo. And in thinking about the contributions of Rosa, one can come up with many things, but really just to summarize some of her sort of key sort of attributes to our understanding of the mechanisms that drive sort of mammalian embryo development, and also the realization of the importance of extraembryonic tissues in the development of the embryo, I think one can sort of summarize a number of her contributions, starting with her outstanding sort of technical sort of prowess in tour de force experiments, fate mapping some of the lineages, her use of chimeras. For example, looking at the formation of the midline shown here in the middle of this embryo, she worked on, she generated chimeras of brachyury mutants, brachyury being one of the first, or in fact the first developmental gene that was positionally cloned in the mouse and thereafter identified in other vertebrate models. Rosa was also in this age where many of us are interested in generating tissue or single-cell specific transcriptomes. Rosa was involved in one of the first, and produced, in fact, the first tissue-specific transcriptomic libraries in collaboration with Hans Lirach for the early mouse embryo. And I think finally, I think her contribution has been to realizing the importance, as Yumiko said, of some extraembryonic tissues. And if I think of Rosa, and in preparing my talk for today, I can think of two words. And for those of you who knew her, those are not cigarettes and alcohol, but visceral endoderm. And really what I thought I would talk about today is some work that has been going on in my lab, a project that's been developing over the past 10 years, really looking at the visceral endoderm. What I thought I will tell you about is our studies trying to visualize cell behaviors in living mouse embryos, studies that have revealed a critical role for the visceral endoderm in the morphogenesis of different tissues that appear at gastrulation. And then I would challenge a sort of dogmatic view of mammalian development which posits the strict segregation of embryonic and extraembryonic tissues. And I will suggest in some preliminary experiments that I think perhaps may be too preliminary to discuss, but I think they're very provocative. And in the vein of Rosa, I thought I would share them with you. And these are experiments fate mapping the visceral endoderm that suggest that a tissue that previously was ignored as being not particularly interesting and specifically extraembryonic may in fact contribute descendants to somatic tissues, at least until late fetal stages, begging the question of whether there is a contribution to the adult mammalian organism. So I just wanted before I start, I wanted to frame everything in the context of the blastocyst. This is the pre-implantation embryo that was introduced earlier in the mouse part of the meeting. And this is effectively a universal stage of mammalian development. So this is a mouse blastocyst, but a human blastocyst looks very similar. A blastocyst comprises 3 cell types: the epiblast, trophoblast, and primitive endoderm. And effectively what I'm doing here is color-coding the 3 lineages of a 3D reconstruction of a mouse blastocyst. It takes 3 to 4 days to form a blastocyst in mice and about a week in the human. And I'm showing a late-stage mouse embryo with, again, color-coded lineage contributions. So effectively, the blastocyst involves the specification and stereotypical position of 3 lineages. The pluripotent epiblast will, according to every textbook, give rise to all somatic cells, whereas the trophoblast and primitive endoderm will give rise to the fetal portion of the placenta and extraembryonic membranes. So towards the end of my talk, I'm going to challenge this dogmatic view of this strict segregation of mammalian lineages by suggesting that perhaps descendants of the primitive endoderm give rise to somatic descendants. So when I think about Rosa, and harking back to the early embryo and the blastocyst, I've just sort of drawn this cartoon to bring everyone sort of up to speed and to frame some of her contributions. One can see that by early post-implantation, before the onset of gastrulation in mouse, the general apposition of tissues that are established in the blastocyst are maintained, except there is massive cell proliferation. So the epiblast, shown here in red, sits adjacent to a tissue that's derived from primitive endoderm, shown here in blue, and this will really be central to my talk today, and this is the visceral endoderm. And over here is the trophoblast derivatives in the extraembryonic endoderm. If I think about what Rosa's work and the work of her lab have sort of informed us, is that really the successful mammalian development is critical and dependent upon crosstalk between these tissue layers. And the mouse is really unique in that, or mammals are unique, and they're distinct from many of the other multicellular mammalian or vertebrate and invertebrate systems that have been discussed in this meeting, in that mice are not pre-patterned. And the break-in symmetry that dictates the anterior versus posterior axis in the mouse embryo comes from specialized movements taking place within the visceral endoderm. And I just wanted to highlight some of the key papers, because really this year is the 15th anniversary of Rosa's untimely passing. And were she to be around today, this would also mark her 60th birthday. So really, Rosa was instrumental in demonstrating this asymmetric expression of markers within this visceral endoderm. And this is really, I think, the paradigm-shifting paper that was published 20 years ago in November that demonstrated that this long ignored tissue, the visceral endoderm, in fact in this paper she calls it the primitive endoderm, harking back to the blastocyst, this gene, the asymmetry of this gene precedes the onset of gastrulation. And thereafter she and many of her colleagues demonstrated the earlier asymmetry, and this is a gene that I think many in the audience are familiar with, the hex gene, another transcription factor, that really demonstrated early asymmetries in this extraembryonic tissue. In a sort of very simple-looking but technically rather difficult fate mapping experiment, Rosa style, she demonstrated where she could sort of label cells at the tip of this 5.5 embryo within the visceral endoderm would sort of translocate to this anterior region, and then later using transgenic reporters that were developed by Tristan Rodriguez, who is here in the audience, and in collaboration, or in a long-standing collaboration between Tristan and Shankar Srinivas, they demonstrated by 3D time-lapse live imaging that this position, or these cells in the sort of distal visceral endoderm would translocate to an anterior position. And a lot of work has been done characterizing this behavior that we now know is a very unique collective cell migration within the plane of an epithelium. So effectively, just to summarize, and this is a cartoon of Rosa's from a sort of review authored by Rosa and her collaborator, colleague and close friend Liz Robertson, really Rosa's work and the work of her many colleagues and collaborators set the stage and brought about the realization that it's extraembryonic tissues that bestow upon the embryo its early pattern and its break-in symmetry. So what I want to do today is to take over sort of where she left off and to talk about the role of this extraembryonic tissue during gastrulation and the morphogenetic movements that we're starting to characterize in the mouse embryo. And I wanted just to bring everyone up to speed to just highlight, this is the blastocyst, a pre-gastrulous mouse embryo, and a mouse embryo at gastrulation. And I just wanted to highlight that gastrulation does a number of things, but effectively what it does is transform an embryo comprising 2 cell layers into effectively 3 cell layers. Effectively, the epiblast will form the ectoderm, mesoderm, and endoderm layers at gastrulation. The mouse embryo is perhaps a little unusual in that it's cup-shaped, but effectively, other than that, it looks very much like a chick. If one thinks about gastrulation at sort of leading up to the onset of gastrulation, you see the appearance of a structure that marks the posterior of the embryo. The appearance of this primitive streak is subsequent to this anterior position within the visceral endoderm of this, what we now call the anterior visceral endoderm. And effectively, during gastrulation, cells of the epiblast will move into the primitive streak. There they will undergo an epithelial to mesenchymal transition, and then they will migrate away as wings of mesoderm. This is just a confocal sort of image of a transverse section of an embryo at this stage. This is a scanning EM that highlights the morphology of the different layers of cells. The epiblast, which is a columnar epithelium, the mesenchymal mesoderm, the direction of its migration, and this outer squamous epithelium, the endoderm. And I just wanted to highlight that this is a marker that Rosa really brought to prominence, at least in the mouse, brachyury. That this is a reporter that my lab have just generated, which is a transcriptional readout of FGF signaling, and this is a postdoc who's currently characterizing this reporter in my lab, Sophie Morgani. So Sophie's actually, and the reason I thought I should mention her specifically at this point, she's a scientific granddaughter of Rosa Beddington. That in fact, she performed her PhD studies and was a graduate student with Josh Brinkman, who's formerly a postdoc of Rosa's. So, and I just wanted to highlight, these are just exploded views to show you that as gastrulation proceeds, these wings of mesoderm will circumnavigate the space between the outer visceral endoderm and the inner epiblast. And what I'm going to talk to you today is about some studies that when I started my lab, I never thought I would be working on. What I wanted to do was to try and visualize cells in living mouse embryos to understand the cell behaviors that drive gastrulation, something that I think Rosa herself was very interested in. And I just want to mention that this was, it took a long time to set up. And effectively what we've done is optimize methods for culturing mouse embryos on the stage of microscopes and in 3D time-lapse data acquisition, as well as the generation of reporters. And what I'm going to talk about today is a project that really came about through the generation of reporters, the acquisition of time-lapse images, and the findings that were not compatible with our sort of expected views of development, and I'll sort of elaborate on that. So effectively, what I just wanted to mention is that the gut endoderm is a tissue that many of us recognize as being internal, running the anterior-posterior length of the embryo at mid-gestation. But what I'm going to talk about today is how it forms at gastrulation. And when it forms in the mouse, it forms on the surface of the embryo. So it's actually rather a nice tissue to be able to have visual access to. So this is when we'll be looking at a lot of embryos at this stage, looking at the surface of these embryos. And in considering gastrulation and the formation of endoderm and how it ties in to the morphogenesis of mesoderm, that the general view was that as cells sort of came through the primitive streak, they would sort of migrate away as mesodermal wings, and then at some point, as a gastrulation was underway, cells at the anterior streak would be specified as endoderm, would insert into this overlying epithelial sheet, the visceral endoderm, shown here in blue, that this definitive endoderm layer that was epiblast-derived and embryonic would somehow expand and displace this extraembryonic sort of visceral endoderm layer to these extraembryonic regions. So one of the reporters that we decided to make when I first established my lab was a reporter for the visceral endoderm. And really what this was meant to do was act as a counterstain so that we can visualize the progression of gastrulation. We really weren't that interested in the behavior of cells that comprise the visceral endoderm. But very soon we noticed that something was going on that was very unexpected. So this is one of the reporters that we generated, and this is just conventional GFP hooked up to the 6 regulatory elements of the alpha-fetoprotein locus that were first characterized in Shirley Tillman's lab. This is a Z-stack demonstrating the specificity of the reporter, and this is one of our first time-lapse images that really suggested that something very different was going on. And I hope you notice that by contrast to our expected displacement of visceral endoderm, what we noticed was that cells would stay put and they were dispersed. And our original expectation was that this was just an artifact, perhaps, of the reporter, so we made additional reporters, and these are just the transthyretin elements that were originally characterized in Terry Van Dyke's lab, driving a red fluorescent protein, and you can see a comparable distribution in stage-matched embryos, and these are just double transgenics, demonstrating that we have overlap. In any type of visceral endoderm reporter that we generate, we identify these cells. And why do we do so? And this is just transgenic embryos, and I draw your attention to this embryo here, which we've live imaged, and then we've processed for an mRNA in situ hybridization. You can see that GFP is localized to this proximal extraembryonic region, but the transcript, but the protein is localized overlying the epiblast, suggesting that GFP acts as a short-term lineage tracer in these embryos, and this is why we can identify these cells. If we perform a fate mapping experiment where we can hook up these AFP or TTR elements to a Cree, we see the same distribution of cells. And I should point out this is a Rosa-based LAC-Z reporter that was developed in Phil Soriano's lab that many of us here have used over the years. I also wanted to mention, or take this opportunity to mention, that if we generate tetraploid chimeras, and tetraploid chimeras are often used as a way of generating completely ES cell-derived embryos. If we generate tetraploid chimeras, and if we label the tetraploid compartment with a red fluorescent protein tag, we notice that there are dispersed cells of visceral endoderm origin, so these are tetraploid cells overlying the embryo, further demonstrating that this is a bona fide distribution of visceral endoderm descendants at gastrulation. And I just wanted to go in with markers and to show you these are 2D optical sections, 3D reconstructions, whole mount views of markers of the visceral endoderm, and this is just one representative marker, HNF4 alpha. These are close-up views of the regions that are boxed, and what you'll notice down here is that the marker is being downregulated in GFP-positive cells that are scattered and overlying the embryo. So effectively, what these data led us to conclude is that the nascent gut endoderm comprises cells of 2 distinct origins. As expected, there are epiblast-derived definitive endoderm cells, but also visceral endoderm cells. And also it begs the question of whether there's a lineage relationship between an extraembryonic tissue, the visceral endoderm. This is a tissue that really we have Rosa Beddington to thank for bringing to prominence, and somatic tissues of adult mammals. And this is just a sequence of embryos, they're not to scale, from the primitive endoderm of the blastocyst through to a mid-gestational embryo, demonstrating the contribution and localization of visceral endoderm descendants. I'll talk about our studies addressing this question first, and then finally conclude with some of our recent and ongoing work addressing this question. So I just wanted to summarize our observation of the distribution of scattered visceral endoderm descendants on the surface of an embryo. These are whole mount views. These are just schematics of sections where we can see the epiblast, the mesoderm, and then the endoderm. And the endoderm comprises cells of both red or epiblast origin and also visceral endoderm origin. And what we wanted to or what we hypothesized, is that cells must insert into the visceral endoderm, and we sort of wanted to see whether cells of the endoderm or cells ending up in the endoderm would be present or take a similar trajectory to the mesoderm during gastrulation. And to do so, we developed an imaging protocol whereby we could take transgenics, we could electroporate them to mosaically label cells within the primitive streak, and follow individual cells in 3D time-lapse during gastrulation. So this work was performed by Manuel Viotti and Sonia Novochin in the lab, and effectively what you can see here is a mouse embryo. Mouse embryos, as I've mentioned, are cup-shaped, so what we do is we computationally reconstruct them so that we can look internally. This is the primitive streak where we've labeled a small cohort of cells. That's the surface of the embryo, and here we're looking internally. And if we actually play this movie over time, and this is a 5-hour time lapse, you can see cells sort of moving away from the primitive streak. And if you're right up front, you'll see that they undergo a shape change, coincident or that could correlate with the MT, and they're sending out anterior oriented projections. To sort of look at our model of cells originating in the streak, being carried through the wings of mesoderm, and to ask whether they end up on the surface, we can image from the surface. And then again, we can see cells that are sort of scattered red fluorescent that appear in these regions where the visceral endoderm disperses. So in instances where we can make out the sort of arrangement of cells within the visceral endoderm, at the point of cell intercalation, we notice something very interesting. This being that cells will always intercalate into this pre-existing epithelium from the mesodermal layer at the vertex of multicellular rosettes. We know from the work of many labs, including my colleague Jennifer Zalen, that rosettes are thermodynamically unstable. So that would argue that perhaps rosettes, when they're interrupted, will collapse and allow space for cells to come to the surface. This sort of event is very comparable to the radial intercalation event that was recently characterized in John Wallingford's lab. Also, the reverse of many of the cell extrusion experiments that have been characterized by Jody Rosenblatt. So we're currently generating reporters where we can visualize live the arrangement of cells in the visceral endoderm so that we can focus in on this detail a little bit more carefully. So one of the things we first wanted to do is to look at mutants that disrupt gastrulation, to look to see whether the visceral endoderm disperses. And these are just some classic gastrulation mutants that have been characterized by various labs. And I think Brian Saruna's in the back, and he, whilst in Janet Rossant's lab when he was a graduate student, characterized the FGFR1 mutant. And you can see that mutants that disrupt gastrulation, when no cells come out of the primitive streak, also fail to disperse the visceral endoderm. But what we really wanted were mutants that would gastrulate but would affect visceral endoderm morphogenesis. And I just wanted to show a series of embryos. These are just wild-type or transgenic embryos, early, mid, and late visceral endoderm dispersal. These are whole mount views looking at the surface. These are section views where you can effectively see the primitive streak, the wings of mesoderm, and these indentations, early, mid, but not late, where you can see cells appear as though they're trying to poke into this outer visceral endoderm layer. And the white arrowheads identify cells that are trying to come to the surface, whereas the red arrowhead is a cell at the surface. When dispersal is complete, we see a surface layer comprising GFP-positive cells that are derived from visceral endoderm and GFP-negative cells that are derived from epiblast. So what we wanted to do is to identify markers that would uniquely allow us to visualize these cells as they were coming to the surface. And we spent a lot of time looking at markers, looking at transcription signatures, and I just wanted to highlight a network of signaling molecules and transcription factors that are conserved amongst vertebrates that we're looking back into in greater detail and to just talk a little about one of these factors, the HMG domain transcription factor SOX17. So SOX17 labels cells uniquely within the region of the wings of mesoderm before they arrive at the surface of the embryo. So these, we believe, are cells that are destined to adopt or are in the process of adopting an endodermal identity. And if we look at sequential stage embryos, sort of during and after sort of endoderm morphogenesis, we can see SOX17 being expressed initially sort of as cells come to the surface, but also later you'll see all cells on this surface, irrespective of their origin, will express nuclear SOX17. These are the real data. This is the schematic of what the data show us. So effectively what this demonstrates is that cells of the visceral endoderm will change their identity, and there is some type of equivalence that takes place between visceral endoderm descendants and definitive endoderm descendants that are derived from the epiblast. And this is perhaps why many of us have sort of been unable to identify these cells, because by sort of 7.5 days, they're remarkably comparable in terms of the usual suspect markers to epiblast-derived definitive endoderm cells. So SOX17 is a marker of definitive endoderm progenitors, and we wanted to ask, beyond just being a marker, is it required for this event? So SOX17 mutants have been characterized originally by Patrick Tam and others over 15 years ago. SOX17 mutants are morphologically distinct from their wild-type or heterozygous littermates at mid-gestation, around 9.5 days. They, in fact, have endoderm defects in terms of failing to close their endoderm, they fail to turn, and they also have laterality defects reflected by a failure in the correct looping of the heart. 24 hours earlier, you'd be hard-pressed to distinguish a SOX17 mutant from a wild-type littermate. But if you were to throw a visceral endoderm reporter onto mutant embryos, 24 hours earlier than this stage, as shown here, you can see something very profound and very specific. So the endoderm layer on the surface of the embryo in SOX17 mouse mutants comprises exclusively or almost entirely of visceral endoderm descendants. That is shown here. And I just want to contrast with the gastrulation mutants, because these embryos are gastrulating in that they're generating a mesodermal layer. And this is just schematized over here. So this is, in fact, a very specific defect that specifically affects the formation of definitive endoderm progenitors. And in experiments that I don't have the time to show you today, really what is happening in SOX17 mutants is a complete failure of definitive endoderm specification. So one of the things we started to think about when we were thinking about how cells come to the surface and they're turning on a program of an endoderm identity is how do they do so? And then we wondered sort of what is happening at gastrulation in terms of these basement membranes that are present to separate tissue layers. And we realized that in addition to driving the formation or the transformation of an embryo comprising 2 tissue layers to 3 tissue layers, what gastrulation also does is propel the formation of 2 basement membrane from a pre-gastrular stage embryo comprising 1 basement membrane. So effectively what you have at gastrulation are basement membranes at the epiblast mesoderm interface and also at the mesoderm gut endoderm interface. Whereas before the onset of gastrulation, there's a single basement membrane that's positioned at the epiblast visceral endoderm interface. And we hypothesized that this could be accomplished by 2 not necessarily mutually exclusive models, whereby the mesoderm layer, as it started to form, could either bifurcate the existing basement membrane and then reinforce it, or alternatively, it could migrate on one and then deposit a second. Anyone sort of who's thinking about this might realize that I'm a little biased here, that this could take place in 2 orientations. I've specifically drawn it in one orientation, namely that mesoderm migrates on an epiblast, on the basement membrane that lies at its interface with the epiblast, and then deposits a second basement membrane at the interface with the visceral endoderm, because that's what we believe, or that's what our data suggest may be the case. So we've started to look at sequentially staged embryos in whole mount and then in section, and our data would suggest that there is de novo formation of a basement membrane at the sort of a mesoderm-endoderm interface. We're now generating reporters so that we could live image ECM assembly in living mouse embryos. And if we look carefully, we also notice, and this will become sort of apparent later, is that during its morphogenesis, the gut endoderm goes through a transition period where it is sort of multi-layered, and that this layer will resolve into the single-layered epithelium comprising visceral and definitive endoderm cells. If we look at this stage, and we look at cells that are GFP negative, they're of course SOX17 positive, demonstrating that these are nascent definitive endoderm cells. And this is just a cartoon to schematize everything, that effectively we've noted this intermediate period where this multi-layered endoderm epithelium sort of comprises cells that are SOX17 positive and negative, and the basement membrane is starting to be sort of assembled at their putative or prospective basolateral interface. So what we wanted to do is to look at the localization of basement membrane in SOX17 mutants, and we saw something very profound. This is just a laminin A1 localized antibody stain, but we've actually looked with a number of basement membrane components, and we noticed that SOX17 mutants exhibit a complete absence of basement membrane at the mesoderm-endoderm interface. And this is just schematized over here versus a wild type. So one of the things that we sort of immediately reasoned was SOX17 as a transcription factor. Clearly, SOX17 must be regulating the expression of genes, encoding components of the basement membrane. And to our surprise, when we looked for the expression of these genes in mutant embryos versus wild-type embryos, we noted that generally speaking, there was no difference that we could detect. And this was for any of the genes that we analyzed, which suggested to us that effectively transcriptional regulation of genes encoding ECM components can't be the key sort of mechanism by which which results in the absence of this basement membrane seen in or observed in SOX17 mutants. I want to also highlight that these in situs reveal that the onset of ECM gene expression coincident with gastrulation. As cells leave the primitive streak, they start to transcribe genes encoding ECM components. And you can see that ECM gene expression is not observed in epiblast anterior of the wings of mesoderm. And it's really specific to the wings of mesoderm and also the endodermal layer. So really what we've been doing over the past couple of years is entertaining this model, and I'm going to talk a little bit about some of our findings, which really suggests that there must be a different explanation for this absence of basement membrane that we see in SOX17 mutants. So effectively what I've shown you is that nascent or cells originating in the streak that will end up acquiring a definitive endoderm identity will travel along with the wings of mesoderm, shown here, and then they will intercalate into this outer visceral endoderm epithelium. In doing so, we hypothesized that these cells must acquire apicobasal polarity. We also hypothesized that the cells of the visceral endoderm must transiently relax their polarity to allow cells to insert into this epithelium. Concomitantly, we hypothesized that as definitive endoderm progenitors acquired polarity, they might cluster receptors for ECM components on their basolateral surface, and this would drive the focal basolateral assembly of ECM of a basement membrane. And in doing so, this would marry the formation of the endoderm on the surface of the embryo with its segregation from the intervening mesodermal layer. These experiments are being carried out by a cohort of people in the lab, and I'm going to talk about some of their findings over the next couple of slides. So effectively, if we look at the acquisition of polarity, we've looked at several markers, but I just wanted to highlight this. We're looking at the sort of fluorescence at membranes in inner cells versus outer cells, so it's cells close to the surface or sort of internal of the embryo, and we can see that there is a polarized localization of ecadherin, indicative of, in these sort of ingressing cells, indicative of the acquisition of polarity. By contrast, cells that are inner do not have this polarized localization, and of course, as expected, cells, irrespective of inner or outer position, do not have any polarized distribution of ecadherin in SOX17 mutants. Concomitantly, we notice, and this is just looking at integrin alpha 5, the major receptor for fibronectin that's expressed in embryos at this stage, we see a basolateral localization of integrin alpha 5 in cells that are positioned adjacent to the visceral endoderm on the surface of the embryo. Again, by contrast, cells that are internal do not exhibit this polarized distribution of integrin alpha 5. And of course, cells in SOX17 mutants do not exhibit any polarized distribution irrespective of their position. This sort of is really sort of helping us sort of build this model. And what we wanted to do next is to look at some mutants that disrupt components of the extracellular matrix. And we looked at mutants in integrin alpha 5 and fibronectin. And what we noted, to our surprise at the time, was that we can see some dispersal of visceral endoderm in these mutants. I have to say, I was surprised when we first made this observation, but you can see things aren't looking entirely normal. If we take, rather than whole mount views, and we look in sections, we notice that there's a very specific defect. Namely, the endoderm layer in these two mutants is stalled at this multi-layered intermediate. It fails to resolve. And this is sort of shown here. This is just looking at fibronectin in an integrin alpha 5 mutant, a surface view en face showing its distribution, and here a transverse section. So what you'll notice here, in this integrin alpha 5 mutant, fibronectin is not assembled at this junction between mesoderm and endoderm. This therefore suggests that the failure to assemble a basement membrane, even though you're specifying endoderm cells in these integrin alpha 5 mutants, causes a failure to resolve this multi-layered intermediate step into a single-layered endoderm epithelium, and suggests that perhaps the sort of a mechanical input into sort of a resolution of this multi-layered intermediate. So effectively what these data sort of starting to support is a model whereby we have the initiation of a definitive endoderm program by cells that are positioned within the so-called mesodermal layer, the modulation of apicobasal polarity, that should be a bipolarity, in definitive and visceral endoderm cells, the integration or regression of definitive endoderm cells into this pre-existing visceral endoderm epithelium, including the basolateral assembly of a basement membrane and eventual resolution of this multi-layered intermediate into this single-layered epithelium. I've shown you that SOX17 is critical for the specification and execution, or the specification of definitive endoderm cells and execution of the definitive endoderm program, and that genes encoding basement membrane components, for example, fibronectin and its receptors such as integrin alpha 5, are critical for assembly of this basement membrane, specifically at the endoderm-mesoderm interface, and not at this interface, the epiblast-mesoderm interface, and that failure to lay down this basement membrane causes a cessation of endoderm morphogenesis at this later stage. So what I want to do in the last couple of minutes is to sort of move away from our studies trying to understand mechanistically how the endoderm forms to asking the question of lineage. Effectively, what happens to these visceral endoderm cells? If we take embryos and we time-lapse image them, and this is just a movie that plays on the loop, there's the GFP sort of 3D reconstruction in bright field and then in dark field, and we're highlighting cells in the dark field portion of the movie that are dividing. So effectively what this movie demonstrates is that visceral endoderm descendants persist and proliferate after endoderm morphogenesis, or this intercalation event, is complete. And what you'll also notice, and I think this is very interesting, is that these cells are part of an epithelium. They have apical junctions, but as you can see, they're very active and they're sending out basolateral projections. In fact, if you stare at this movie long enough, I told you that these cells, irrespective of their origin, will express markers that are signatures of an endoderm identity, such as SOC17, FOXA2, CXCR4. But you'll notice if you stare at the movie long enough that cells that divide, or visceral endoderm cells that divide, the two daughters will not sit next to each other. They appear to be repelled. And this is something that we don't understand at this point in time and is an open question that we're wanting to address. But what I want to spend the last few minutes of my talk addressing is the fate of the visceral endoderm. So does it really just provide a scaffold for getting definitive endoderm cells to the surface of the embryo, or do these cells persist? And I should just prelude these data by saying that this is really an ongoing project, but I think it's very provocative. So I thought I would show you some of the data. So really this is the question. It's the dogmatic view of the segregation of the lineages that are specified in the blastocyst, which posits that it's the epiblast that exclusively gives rise to all somatic cells. And the alternative hypothesis that perhaps some visceral endoderm descendants might persist and contribute to some endodermal derivatives. So this is the question framed within the context of the late-stage embryo, and that is really, do cells of the endoderm, the tissues of the respiratory and digestive tracts and associated organs, contain any visceral endoderm descendants? And we can think about this initially at a mid-gestation, around 8.75 or 9 days, and consider the foregut, midgut, and hindgut. We can take our reporter-expressing embryos. These are AFP, GFP embryos. This is just one embryo, but we've actually done up to 5 embryos, and we've sectioned the entire embryo, and we've used software to reconstruct, to count every cell within the endodermal layer, and identify every visceral endoderm descendant. We've used this reporter, but also a Cree reporter and they're in agreement, but effectively what you'll notice is the gut tube at this stage comprises just over 5,000 cells, and around 1,000 of these appear to be visceral endoderm descendants. So if we look at foregut, midgut, and hindgut, you'll see that there really is a concentration of visceral endoderm descendants within the hindgut region. This is the region that will eventually give rise to the colorectal region. This is unfortunately the later stage at which we can perform this type of standard analysis, because anyone that's familiar with the endoderm will recognize that many of these early markers are reactivated in different lineages of the endoderm. So effectively what we need to do is to go to a genetic inducible fate mapping strategy. So we're driving a Cree-RT2 in this case by a visceral endoderm specific promoter. We're pulsing early and we're chasing late. This is a difficult time to pulse because we're pulsing around 5.5 days. And embryos, pregnant females, don't like to receive large doses of tamoxifen at this stage. So it's technically a challenging experiment, but I think the data are kind of interesting. So we've generated 2 visceral endoderm specific inducible Cree lines, and we're using 3 very widely used Cree reporters in combination. We're using the Soriano Rosa 26-lac-Z reporter, the Lee Quinlow MTMG reporter, and also the TD Tomato Cree reporter that was developed by the Allen Brain Institute. And I should just say that generally the data from all crosses appears to agree, and I'll show you just a selection of it. And again, this is an ongoing set of experiments. So effectively, this just shows you a 7.5-day embryo where we've pulsed at 5.5 days, and we're analyzing at 7.5 days. And this is the Soriano LAC-Z-based Cree reporter. And as you can see, the labeling is visceral endoderm specific, but unfortunately, this is not as efficient as a regular Cree, as many labs using inducible Crees are noting. So effectively, what we label is only 10% on average of the visceral endoderm. So that's something to bear into mind, that everything that we see should be multiplied by 5 or tenfold. If we go a little bit later, so this is at 9.5 days, and we look along the antero-posterior length of the endoderm, we can see visceral endoderm descendants in the liver, the midgut, and the hindgut. We can use the TD tomato reporter, again with the TTR Cree RT2, again at 9.5 days, and we see something very similar, the sort of persistence of visceral endoderm descendants to these tissues. And this is just the AFP report, Cree, so it's a different Cree, and again we see something very comparable, a similar localization. Looking a little bit later, we can sort of these are just whole mount views, and we've done the experiment. This is just data from the 2 different Crees and the Allen Brain Atlas tomato reporter. This is the yolk sac. This is our positive control for labeling of visceral endoderm descendants, and that is positive. And we can take these embryos and we can look in section, and you notice at 12.5 days we see descendants in the liver, but also in the midgut. We can look at 16 days, and this is really sort of my final slide, but again, I think this highlights a contribution of visceral endoderm descendants in the liver, in the pancreas, but also in the midgut, also in the hindgut. So I just wanted to finish off with just the sort of open question that perhaps a lineage that was brought to prominence by Rosa Beddington's experiments exactly 20 years ago, almost to the day, may in fact challenge the dogmatic view of the strict segregation of embryonic and extraembryonic tissues. And in fact, the visceral endoderm may contribute descendants to somatic tissues. I think I'll stop there. I just wanted to thank the people in my lab whose work I sort of talked about today. And I think I mentioned most of them during the talk. I wanted to also mention that I didn't have time to talk about, but we have a sort of very fruitful collaboration with Liz Robertson, a colleague, friend of Rosa Beddington's, and we've been working with Liz to look at endoderm morphogenesis in various mutants. And of course, this being sort of a mouse genetics meeting, I really wanted to thank the many colleagues that have made available to us many mice that we've used in our studies. And I think I'll stop there. I'll thank you for listening, and I'd be happy to take questions.
Okay. Thank you very much. So, question, please. Go ahead.
Does the transcriptional profile of these persistent visceral endoderm cells in later stages, is it different from the surrounding cells?
Actually, that's a very good question, and I don't have an answer for you because those experiments are about to start. So I think those are really critical experiments because every standard endodermal marker that we and everyone else in the field uses identifies both populations irrespective of their origin. And I think it'll be very important, since we visualize distinct behaviors and cells seem to avoid one another, that we determine whether there are transcriptional differences between cells that are based on their origin. So we don't know at this point.
Great, thank you.
Go ahead.
So if you try to genetically ablate those visceral endoderm cells, do you think they'll be replaced by endoderm cells?
That's actually a very good question. So we now have the inducible Crees working. They're not very efficient. And what, in principle, we could do is use one of the Rosa-based toxin-expressing or conditionally activating toxin-expressing strains to ablate these cells. We don't know at this point in time. But it's a good question. My guess would be that they may not be critical, but we have to do the experiment.
Do you think there are clinical consequences for this mosaicism?
Well, one thing, and I see Jeannie Lee at the front, and well, one thing that I didn't mention is that the visceral endoderm exhibits imprinted exon activation. And we need to look to determine whether that exon activation status is maintained later, because that would suggest there are some molecular differences. I don't know at this point in time whether there are clinical consequences. But what you've seen that the fate mapping data suggests that there are descendants of visceral endoderm within the hindgut, and in fact. Oh, okay. My slides have disappeared, but they see that. So, okay. So there may be. We can discuss it later because.
So, in a tetraploid aggregation experiment, do you see polyploid cells? Have you?
So this is one of the issues with the tetraploid aggregation experiment. So the first is having good ES cells that will take you all the way to birth. The second is the visceral endoderm cells that persist. If you look carefully in tetraploid chimeras, they're larger because they are polyploid. So we haven't as yet done the experiment with a good ES cell line will give us embryos that will go all the way to birth to start looking at their endoderm. And that is something that perhaps we should look at. But I wouldn't be surprised if those cells do not behave the way normal diploid visceral endoderm cells do, because if you look under a high resolution the way we do, they do look a little distinct.
Okay, no more questions? Okay, thank you very much.